talks. Andrew.
For those of you in the back, there's plenty of chairs up here. You just need to squeeze in.
Okay, so I'll be there and I'll be 3 minutes.
Hi, I'm Andrew Moschetti with Google Cloud's Healthcare and Life Sciences team, and want to talk to you today about a couple of the tools that we've got helping to support some of the open work here, such as Variant Transforms, which is an ETL tool for importing VCFs into BigQuery to facilitate data analysis. So if you're not familiar, BigQueries are scalable data warehouse. The main thing I want to point out about that, I think most people have dealt with this before, but one of the changes in the last year is we now support individual regions for BigQuery. So it used to be a multi-region thing, like it would only run in the EU or the US, and you can actually get that down to a data center level now. So if you have a country where you need to maintain data residency, such as in Netherlands, Belgium, Japan, you can specify those individually and keep your data there. So it does support standard SQL as well to do that analysis. And so we'll take a look here at some of the stuff that Variant Transforms has. So Variant Transforms is written with the Apache Beam SDK. This is a version of it running right here in Dataflow, which is our managed Beam service. But looking at this import here is the 2,500 or so samples of 1,000 genomes and about 360 million variants. About 100 days of compute time, running that here in about 2 hours. And so this, you can see where the graph flattens out there. That's the number of workers I've assigned. So I've capped the workers at 400 for this experiment. But obviously that could be adjusted as needed in the configuration. So primarily I want to focus today on some of the things that we've added since last year when we had first presented about this. So the first being some of the additional support we've done for annotations. So the first of that is supporting new annotation sources. So an example of this, we've done some work with Genomenon in the past year to bring their mastermind cited variance reference. And they're actually using Variant Transforms to do the import of their annotations into a public data set that they then make available. So in addition to using Variant Transforms to just import your own variants, we can also use it to help support bringing annotations in if you have annotation sources that you want to make available as well to other people. And then improving the support for annotating existing data sets. So if you've got an already existing data set, we've improved that performance about 10 times within the past year. So doing a VEP annotation on the Illumina Platin genomes now runs in just over an hour. The next is BGZF support. So obviously a lot of reasons that we would want to compress VCF files. I think everyone's familiar with that. The downside of that when we're importing them is that we couldn't import a compressed file we couldn't shard that import until it had been decompressed first. So while the size was great, the runtime was not very practical with a gzip file. Supporting BGZFs now, we can keep a similar file size and actually shard that import as we read the individual blocks and get that runtime of an import down to be on the same order there of around an hour. And the example here is with the Nomad whole genomes, so 261 million variants and the 23 files for the chromosomes there. And the last part I want to highlight on is some of the containerization work that we've done. So we've removed the dependency with Google Cloud's Pipelines API, although you can still use that if you want. But we now actually package it as a container with the binary and all of the dependencies pre-installed there. We make it available at the repository listed here, but you can also pull the code from GitHub if you want to build it yourself, and then go ahead and run Variant Transforms wherever you'd like. And last, a couple of the changes, just a preview of some of the stuff we're still working on. So we're working with prototyping some new schemas. So as we start to deal with running things like joint genotyping on the order of 50 to 100,000 samples, the current schema is not quite adequate, so we are doing some work there with a number of partners in the space as well to come up with new options. Working on updating our parsing libraries as well to go ahead and get some performance improvements out of that. And then continuing our work with third-party annotation. So whether that's annotating existing data sets or if you're an annotation provider, finding ways that you can use to make that available as well. So thank you very much.
So put your questions online, please, and we'll do them in a moment after 2 more short talks.
Good morning and welcome. I'm Michael Buzinger and I work for Brigham Genomic Medicine program, and in the next 5 minutes I'm going to introduce you to Anfisa, which is our variant curation tool. Well, there are many variant curation tools out there, and I'm pretty sure that developers for all of them will tell you that the tools are easy to use, user-friendly, and use a lot of annotations coming from different sources for variant filtration and variant interpretation. But each tool has something unique to them, and I will talk a little bit about what is unique for Anfisa. First of all, Anfisa supports both clinical and research workflows. Most commercial variant curation tools really focus on clinical guidelines and supporting diagnostics and clinical workflows. In fact, I know only one tool that fully supports research workflow, and this is Seeker from Broad. We support both clinical and research, and moreover, our idea is to use research workflow to develop new clinical rules and new clinical workflows. We also focus on collaboration between teams, and Anfisa is being developed by an international community of developers coming from United States, Russia, and Austria. Anfisa was born out of work on undiagnosed disease, where basically each case, if solved, led to some kind of new scientific work. So we support crowdsourcing, trying to select some unidentified information that can be used by various teams around the world to solve and understand what's actually going on with the patient. Finally, Anfisa applies to genomics techniques that long have been developed for, let's say, business intelligence, and we use them for genomic works. Oh, sorry. As I said, we use Anfisa as an environment to develop new clinical rules. And we represent clinical rules as a decision tree to select variants for a manual review by molecular geneticists. And as a development environment, it should support various things. So first of all, the code is written in Python in a similar way like you use Jupyter notebooks. And then you have a step-by-step debugger where you can see what actually happens with your variants while they're analyzed. Here's an example of a clinical rule that we have developed for a hearing loss project. So first we filter, we start with whole genome, which includes about 6 million variants for trio, and we filter out those variants that do not pass quality metrics, and then we select out of the remaining 5 million something, 70,000 that actually fall into gene panel relevant to hearing loss. The next step would be to look at ClinVar, and here we immediately select those variants that are uncertain, significant, or pathogenic with consensus in ClinVar, and discard those variants for which ClinVar consensus is that they're benign. And the next will go to the next step, which is looking at allele frequency using NOMAD. And again, we immediately select the extremely rare variants and discard common variants, and again, those that fall in between go to the next step. And as I said, at each step, we can actually drill down and look at what kind of variants, for example, we discarded at this step, and we see that there are certain genes, certain most severe consequences, and these variants are present in certain databases, so we can actually look and see what we're doing. So Anfisa is available today as a beta release. You can download it from GitHub, and it is used by ECCB project and by Brigham Genomic Medicine working on UDN projects. And a couple of words about our next steps. We want to develop an annotation service for Anfisa where you can use your VCF and immediately see the results on our website. We integrate with FAVOR database. FAVOR is a comprehensive human genomics database supported by an HGRI grant, and we'll look into integration with Hadoop and support for machine learning. Thank you.
So one more talk, and then questions for the last three.